Oh my gosh, I forgot to include a narration for peroxisomes in the part 2. Well, we'll have one quick little narration on this one slide. So the last organelle that's covered in the endomembrane system section of your textbook, section 4.4, is peroxisomes. And I have to admit to you, it's a bit confusing to me why they've included peroxisomes in this section. And this is the reason why. Peroxisomes do not share pieces of their membrane with other parts of the cell. So per my knowledge and understanding, they are not physically part of the endomembrane system, but they're sort of standalone organelles. Mitochondria and chloroplasts are also standalone organelles that aren't connected to the endomembrane system, but though they have their own DNA inside of them that sort of controls what they're doing to some extent. Peroxisomes do not have their own DNA inside of them, but yet they're still kind of autonomous or separate from the goings-on of the rest of the cell. So with that being said, let me just quickly talk about what peroxisomes are and what they do. They're very small, sort of spherical organelles. They would probably look kind of like a vesicle, but they have a very specific job or function. And what they do is they confine destructive chemical reactions away from the remainder of the cell. So specialized little compartments for chemistry that otherwise might damage a cell if that chemistry was done in the cytosol. So the peroxisomes contain two sets of enzymes. There's about 40 total. One of those sets of enzymes takes fatty acids. Make sure you remember what a fatty acid is, a long hydrocarbon chain with a carboxyl group on the end. We find fatty acids are parts of fats. Anyway, peroxisomes take fatty acids and chop them into little tiny pieces that can then be used inside your mitochondria for energy. It's kind of like taking a tree and chopping it up into pieces of firewood that you can then use inside your, fireplace or wood stove or whatever to burn. Otherwise, the tree's too big to burn. So fatty acids would be too big to process, essentially. So they have to be chopped into little pieces. The other set of enzymes inside peroxisomes are used to chemically modify harmful molecules that your cells produce, and by removing hydrogen atoms from those molecules. And in the process of removing those hydrogen atoms, a chemical substance called hydrogen peroxide is produced. Now, this chemistry is confined inside the peroxisomes because hydrogen peroxide is dangerous. It's toxic to cells. So if this chemistry was going on in your cytosol, the fluidy part of your cells, that hydrogen peroxide would do damage to your cells. So eukaryotic cells confine this chemistry to peroxisomes, producing this hydrogen peroxide as molecules are kind of detoxified, but then they have to get rid of the hydrogen peroxide. If it built up and built up, that would be dangerous. So there's an enzyme inside the peroxisomes called catalase, and catalase has a very specific job of breaking down the hydrogen peroxide into water and oxygen, which are completely harmless to the cell. So in summary, peroxisomes, while they are included in the section on endomembrane organelles, I don't think they're really part of the endomembrane. But regardless, you need to know what they are and what they do. And that's what this narration talks about. Little spherical organelles that have enzymes in them for either chopping up fatty acids to be used as an energy source, or chemically modifying molecules to make them less dangerous, but then producing hydrogen peroxide that has to also be broken down.